Okay, welcome to session 2. Today, we cover newly introduced technologies and their application. We have 3 speakers from Korea and Hong Kong and China. So I'm going to start with the first speaker, introduction of our first speaker from Korea, Dong-Sung Lee. He is from University of Seoul. He introduced about some his epigenetic technique for the single cell analysis. Please welcome Dr. Lee.
Good morning, everyone. I'm Dong-Sung Lee from University of Seoul. It's a tremendous honor to present here at Human Cell Atlas Asia 2023. I'd like to start by expressing my deepest gratitude for this opportunity. I've been studying genome and epigenome within our bodies. Genome is the blueprint, and epigenome dynamically changes and indicates cell fate even with the same DNA content. This includes DNA methylation, histone modification, and chromatin conformation. We've been conducting research using next-generation sequencing, which simply is a technique for reading DNA sequences. NGS has evolved beyond just genome analysis to include profiling of various features, including transcriptome and multiple epigenetic features, including DNA methylation, protein binding, chromatin accessibility, and chromatin conformation. And all of the techniques have further developed to be applied at the single-cell level, and more recently, single-cell multimodal omics techniques have emerged which can profile multiple cellular features with one experiment. For instance, we can profile both transcriptome and cell surface proteins simultaneously at a single-cell level with CySig. And other techniques have been developed to simultaneously profile more than one features. We have developed single nucleus methyl-3C or SNM3C, which can simultaneously profile DNA methylation and chromatin conformation, and apply this to human brain in 2019. I'll briefly talk about the experimental aspect of this. We first do in situ. I'll briefly talk about the experimental aspect. We first do in situ chromatin conformation capture. We basically cross-link the cells, digest them, and re-ligate them. By doing this, what we expect to see is that DNA is folded inside the nucleus. We expect to have higher chance of ligation in the sites that are at the close proximity. We do this on a whole genome scale, with the whole genome scale, so we can measure the distance of every point on the genome. With the data obtained in this way, we are able to confirm the 3-dimensional structure of the genome. And this capture process is all done without breaking the nucleus membrane, so we can sort and isolate the cells in each well of the well plates. After sorting, we extract DNA inside the well and treat them with bisulfite so we can retain the DNA methylation information. And then we mark the wells with the unique barcodes and do the sequencing. Initially, to test whether this method can actually distinguish different types of cells, we experimented with stem cells and mammary gland cells. In this case, we conducted a blind test of analysis, knowing the answer. As a result, we were able to confirm that both DNA methylation and chromatin conformation data could almost perfectly differentiate the types of cells. After applying SNM3C to the human prefrontal cortex, we obtained data from 4,238 single cells and were able to identify 14 cell types. The most important point that I'd like to emphasize from this data is that we were able to identify cell types using DNA methylation information. While clustering is possible with chromatin conformation, cell type identification was not feasible because there was no existing reference from studies of various cell types using chromatin conformation. And of course, we are currently also conducting analysis for cell type identification using chromatin conformation also. The heat map you're currently viewing represents each row as individual single-cell data, and each column shows CpG methylation at base resolution. You can see that the characteristics of each cell types are clearly evident even at the single-cell level. To simplify the analysis, we aggregated the data with the identified cell types. Similarly, a high level of data can be obtained for chromatin conformation at the single-cell level, and likewise, the data were combined by the types. What we learned through the initial research is that both DNA methylation and chromatin conformation have cell type specific aspects, and there is a strong correlation or association between the two. The 89,356 differentially methylated regions and 36,559 differentially interacting regions we found overlap by 80%. This means that where interaction changes, methylations also changes, and vice versa. Additionally, there's a strong anti-correlation between the two. In other words, where interaction is strong, methylation is low. And we can see the really strong relationship between methylation and interaction without any statistical test to this. One thing that I would like to emphasize the most with the method is that even though the biggest strength of our method is that we can profile methylation with chromatin conformation, even without that strength, our method has the highest efficiency in the chromatin conformation capture capability alone. Because of this reason, we could generate the data with the highest quality up to date. With the developed SNM3C, we are now participating in the Brain Initiative Cell Atlas Network project. We are specifically focusing on the epigenetic changes that occur during the development of brain. We applied the SNM3C across diverse stages of human brain development, which include mid-gestation, which is comprised of gestational week 18 to 23, and late gestation, which is comprised of the gestational week from 35 to 39, infancy, and adulthood samples. This study is the first single-cell chromatin conformation of DNA or DNA methylation study performed for human brain development. We identified a total of 139 cell populations by a fusion of 3 data modalities, which is non-CpG methylation, CpG methylation, and chromatin conformation. These cell types are organized into 10 major groups, and what we could see from this data is, first, excitatory neurons had distinct epigenomic types in the human prefrontal cortex, between the human prefrontal cortex and hippocampus, whereas inhibitory neurons are broadly shared between the 2 brain regions. And the second observation, globally, what we could see was that neuronal cell types, astrocytes and oligodendrocytes progenitors, were strongly separated by developmental stages, whereas oligodendrocytes and other non-neuronal cell types showed similar epigenomic patterns across development. The developmental increase of non-CpG methylation in neuronal cells is an epigenomic hallmark of neuronal maturation. What we could see by dissecting the cell types is that accumulation of non-CpG methylation in hippocampus neurons occur earlier than that in PFC neurons. And these dotted lines show the methylation changes in HPC, whereas these continuous lines show the changes in PFCs. And the changes in HPC occurs earlier. Although genome-wide CpG methylation levels only show moderate dynamics globally, Clustering of gene body CpG methylation readily separates PFC neuronal populations into prenatal and postnatal groups. A similar clustering analysis of HPC neuronal population found that the remodeling of gene body MCG occurs in HPC between mid-gestation and late gestation. The majority of neuronal populations in the late gestational HPC were grouped with infant and adult cells rather than with mid-gestational cell types, suggesting that the maturation of these cells in HPC were done before late gestation. We also tested individual loci. All of the selected genes that show a gain of methylation, here on the right side, gain of methylation during development underwent MCG remodeling in HPC ahead of PFC, as well as the vast majority of genes showing developmental loss of MCG showed earlier change in HPCs. Together, we found the remodeling of non-CpG and CpG methylome predominantly occurs between late gestation and infant stages, starting in the HPC and proceeding shortly thereafter in PFC. Cell type classification based on DNA methylation and chromatin conformation are largely concordant, with DNA methylation profiles providing a greater resolution for cell type classification in general. However, we found a notable exception in mid-gestational brains where a single neural progenitor radioglia population defined by DNA methylation signatures can be further divided using chromatin conformation signatures. Using chromatin conformation, we grouped RG cells into neurogenic population RG1 and a putative astrocyte progenitor population RG2. This observation led us to speculate that chromatin conformation dynamics precede the remodeling of DNA methylation during the differentiation of astrocytes. To test this hypothesis, we computed pseudotime scores for RG1, RG2, and astrocyte cells using either gene body methylation or the interaction frequency of genomic bean pairs. The research suggests little CpG dynamics in RG1. As you can see, the RG1 and 2 are overlapped to each other, and RG2 population, and substantial remodeling of MCG in differentiated astrocytes. In contrast, the reconfiguration of chromatin interaction was more continuous across the differentiation of RG to astrocyte, which resulted in a drastically different distribution of pseudotime scores computed from CpG methylation or chromatin interaction. Consistent with genome-wide pseudotime patterns, gene-specific analysis found the remodeling of interaction gene scores generally occurs in RG1 and RG2 population and precedes MCG dynamics in differentiated astrocytes. And we could see that this chromatin conformation preceding happens like globally in the differentiation of other cell types too. We also have found a new layer of unique epigenomic regulation in neurons. That is the unusual domain-dominant chromatin conformation that is different from the compartment domain conformation in non-neuronal cells and non-brain tissues, suggesting neuronal specific regulations of cohesin-dependent enhancer promoter interactions. As in the research of other groups in 2017, we basically analyzed global interaction patterns. In fact, the analysis was started with the expectation that the cell cycle could be observed by doing this analysis. However, our observation indicates that this global interaction changes does not appear to indicate the changes in the cell cycle but can be used as a strong marker to distinguish the cell types too. And this is the paper that I just mentioned, and which was published in 2017. And what they did was basically did the same kind of analysis that we have done, which I just showed you, and claimed that this global pattern represents the cell cycle, which was not the case in our analysis. And our study also presented compelling research for demonstrating that the combination of single-cell Hi-C and DNA methylation profiling is a powerful approach for dissecting the molecular mechanism in disease-associated GWAS loci. This is one example that there is a cell type specific interaction right there, cell type specific interaction and differential methylation occur in one cell type with overlapping with high risk locus we got from schizophrenia GWAS. This indicates the potential power of our approach that would be able to find the target gene, which is RORB in this case, that is affected from the potential enhancers we identified from GWAS. And this interaction could be observed from certain cell type. In summary, we have developed a new technique, SNM3G, which can profile DNA methylation and chromatin conformation at a single cell level and examined developing human brain tissue. We could see a lot of things that are previously impossible to see and found aforementioned research. And above all, I would like to emphasize one thing, that the new technique can lead to new discoveries or new findings. And on this point of view, currently we are focusing on expanding our method to transcriptome to better integration with the single-cell RNA-seq data and also to obtain new findings. Before finishing the talk, I'd like to thank all the people who I have worked with, and thank you very much for listening.
Thank you. You can take a question from the floor or online.
Very exciting to see Dong-Sung. Now that you can annotate the cell types, that's really transformative. You got chromatin conformation for annotated cell types. I was intrigued by your conclusion that the chromatin conformation remodeling happened before the DNA methylation remodeling. Just suggesting, to play devil's advocate, an alternative scenario where simply the DNA methylation data were too noisy to separate those two cell states, the radial glia 1 and 2, if I remember correctly. And the chromatin conformation could separate them, but that doesn't mean that the one modality happened before the other. It could just be, maybe the audio, but it could just be noise.
Sorry, the speaker was not on, so I couldn't hear you very well.
Okay, just a short question. The fact that DNA methylation couldn't separate RG1 and RG2, could that be simply just noise in the data?
That could be what it is, but we also could see the same kind of phenomena from the other cell types too. We have a lot of evidence that shows the chromatin conformation precedes the methylation change. And actually, in the previous study that we have done, which was the methylation bisulfite sequencing done during the reprogramming process of iPS cells, what we could see was that we could see that the transcription factors bind to the genome first, and the methylation change occurs a little bit after that. And chromatin conformation is basically chromatin conformation basically occurs with the proteins. So I think this data kind of makes sense, and which was predictable.
All right, so you have independent evidence. Thank you. Really powerful assay. Congratulations.
Okay. Thank you very much.
Next, Jay, and then short questions.
Really cool work. Congratulations again. You use DPN2, which is a fairly low resolution restriction enzyme. Any thoughts on improving the resolution so that you can see promoter enhancer and cross.
Yes, we've been testing the like other digestion approaches too, like MNAs and stuff. But what we actually want from this single cell assay is the like high efficiency of ligation. But if we start to use like different like approaches, then those are gonna start to make the like blunt edge like and lower the like efficiency. So we're just like keep like trying like different things right now. Maybe uses more number of restriction enzymes, but yeah, I mean, we are pretty much satisfied already though.
Or maybe combinations of different restriction enzymes.
Yes, yeah, we are also testing that.
Cool, thanks.
Great, thank you.
Okay, from online questions.
Yi from GIS says, great talk. Have you tried combining single nuke M3C and single nuke RNA-seq to look at chromatin conformation and gene expression dynamics?
Are you asking like if we have like used the SNM-CT and SNM-3C to combine the data together, or like just the ordinary single-cell RNA-seq data?
Hard to say. So answer how you feel is
combining
best.
the RNA-seq data? Yes, and actually that's what we're really striving for right now, trying to combine the RNA and transcriptome to the data as a method, because like as most of you are studying single-cell RNA-seq right now, so it will give us a lot of like in-depth like insights onto the biology. And so basically, short answer is that we're making it, we're developing it right now, but there is actually a method that we can profile DNA methylation and transcriptome simultaneous already. And we have the method that we can profile DNA methylation and chromatin conformation together, which means if we just use these two together to one sample, we will be able to match the cell types by using the methylation, and so we will be like integrate the data between chromatin conformation and RNA-seq too. So we already have the answer for that, but we still want to develop a new method that we can just profile all of the three like together.
Thank you.
Okay,
Thank you. Thank you, Dr. Lee. Thank you very much for your nice talk. And then we're going to move to the next speaker, as chaired by my co-chair and Maitra.
Thank you. So the next speaker is Dr. Angela Wu from the Hong Kong University of Science and Technology. She will be talking about SCON-seq. Angela.
Okay, great. First of all, I'd like to thank the organizers for this invitation. I was actually very much looking forward to being there in person at this meeting. It would have been my first visit to India, but unfortunately, due to various unfortunate events, I was not able to be there in person. So I still appreciate the opportunity to present my work virtually to you guys. So today I'm going to talk about our method for single-cell multi-omics analysis from frozen tumor samples. And also I want to present some new work that was recently published regarding single-cell spatial omics analysis, if I have time. Just a conflict of interest disclosure, I am a co-founder and shareholder of Omics Limited, which is the entity that is commercializing the technology I'm about to talk about. Okay, so single-cell one-seq is a very versatile one-tube method for single-cell simultaneous whole genome sequencing and RNA sequencing. And I'd like to share with you that this work was done by a group of people, including my very talented postdoc, Dr. Yu Lei, and collaborators at Chinese University of Hong Kong and also my home university, Hong Kong University of Science and Technology. So as the previous speaker has already elaborated, single-cell multi-omics analysis is very informative for understanding biology, and I would like to emphasize that it is very informative for understanding cancer. If we look at this data from the TCGA Atlas, where each row represents data from one type of tumor, we'll actually see that the correspondence between DNA copy number changes in this column right here and, let's say, the transcriptome amplification or reduction is actually not as strong as we would think. So, for example, we can see areas where the copy number change is clearly depleted, but in the transcriptome profile, it's not obviously depleted, or it doesn't seem to be as big of a difference. So obviously, this is because of epigenetic regulations between the DNA level and the RNA level, and so on and so forth, as the previous speaker also very nicely demonstrated. So therefore, even though there were earlier works that sort of proposed, we could infer the copy number changes of a cell based on just the single-cell RNA-seq information, we want to argue that actually it may not be a sufficient way to assess copy number variations at the single-cell level. So we really want to have a way to directly measure both the transcriptome and the whole genome information, in particular, the copy number alterations. To give an example of where this kind of heterogeneity can be very important in terms of biology and human disease, gliomas are a deadly and highly heterogeneous tumor type. They are the most common brain tumor that we find in humans, and also typically gliomas have a very low survival rate with few therapeutic solutions. So for example, a grade 4 astrocytoma, the 5-year survival rate is only about 5%, and actually the survival rate hasn't changed very much over the past decade or two, despite our efforts to further understand this tumor. And so on the left, I'm showing sort of the roadmap that clinicians will take, which is a lot of it is based on the sort of genomic profile of these tumors. Some of them include very large copy number changes or chromosomal changes. And as you can see from this roadmap, the genomic variability or the genomic changes that occur in this type of tumor are many. And so at the genomic level, it is a highly heterogeneous tumor. Very recently, there was a paper published in 2019 in Cell, which did a pretty comprehensive profiling of the single-cell RNA-seq landscape of gliomas. And it was found that at the RNA level, the transcriptome level, this tumor is also very heterogeneous. It involves several different subtypes of tumor cells, which resemble different types of normal brain tissue, such as astrocytes or neuroprogenitor cells. So as you can see, at both the transcriptome and the genomic level, gliomas are very heterogeneous. And it sort of is in our interest to understand this heterogeneity if we really want to tackle this problem of poor therapeutic solutions for this particular disease. So our method, single-cell one-seq, really allows us to do that using a pretty simple method. It's a one-pot reaction, meaning that what you do is you sort the cell into a container like a well or 384-well plate or other kind of container. And then after the cell is in there, we just have to keep adding reagents without having to do any purifications or sample splitting in the middle. And at the end of this reaction, what you get is a combined library that labels both the transcriptome and the genomic information from that same single cell. And you can put that library into your sequencer and then bioinformatically back out or back calculate which reads came from the transcriptome and which reads came from the genome. So in the interest of time, I'm not going to go into the details of how this is done. This is published, so if you're interested, you can ask me later or look at our publication. But just believe me that we can do that. Okay, so using this method, we profiled a particular patient that had 3 instances of astrocytoma. There was the primary instance of the tumor, the first relapse, and then the second relapse after multiple resections. And the second relapse tumor was the one that we profiled using our single-cell one-seq method. We took this tumor sample, which actually had been frozen in biobank for 2 years before we got it. And when we got it, it was just frozen brain tissue, which we then processed into a single nuclei suspension and then sorted that into plates for processing. So as I mentioned, because our method is a one-pot method and it doesn't require separation of the nuclei and the cytoplasm to process the DNA and the RNA separately, it can be applicable to frozen tissue samples, which is not often the case for sort of many multi-omics whole genome and transcriptome methods. So using this sample, we did both the whole genome seq and the transcriptome seq, and here are the results. So if we look at the clonality of this tumor, you can see on the left using this simple UMAP visualization that we can see 3 clones in the tumor. This major clone down here in yellow is the most dominant clone, and then we have a sort of smaller clone in green, and then a very small red clone over here, and a large group of normal cells that do not display obvious copy number variations in this tissue that we got, in this brain sample that we got. So this is a quick overview of the clonality of the tumor. And then if we look at the details, you can see in this heat map, we can see the copy number variations in these different clones. So in the major clone, which is the largest, most abundant cell type there, we have these copy number alterations, including some that are commonly known to gliomas, such as MYC, CDKN2A, et cetera. And in this smaller clone, the green one, we can see similar alterations as the major clone with some subtle differences here and there, which explains why, distance-wise, they're characterized as being close to each other. But with this red one, which we're calling the 2R clone 1, the number of alterations that is overlapping with the major clone is fewer. And distance-wise, it actually is closer to the normal cells. So this was interesting to us. So we decided to further look at the RNA sequencing information of these same cells. And so just based on the RNA-seq alone, we were able to also cluster out different cell types, including the tumor cells that are all clustered here in the center. We also sorted them into 4 different populations, as sort of classified by that earlier paper that I mentioned. And all the markers sort of check out there. And then from the normal cells, we can see that there are neurons, 2 different types, oligodendrocytes, tumor-associated macrophages, and astrocytes here in gray. So this also all checks out. All the markers look fine. The surprising part comes when we try to overlay the genomic information with the RNA-seq information. So if I were to draw that same UMAP, but now I'm coloring these cells based on their clonality, we can see that the major tumor clone and that slightly smaller tumor clone is in the center. These 2 are the obvious tumor cells that express a tumor-based transcriptome signature. But what's interesting is that the 2R clone 1, that very small clone, actually falls into the astrocyte population, meaning that their transcriptomic signature is actually resembling astrocytes rather than the major group of other tumor cells. So this was very surprising because if we only rely on the single-cell RNA sequencing information, we may not identify this group of cells as bona fide tumor cells, but since we have sequenced their genomes, we know that they have major copy number alterations and are confident that they are actually tumor cells. So knowing that they're tumor cells, we can compare their gene expression signature with the true normal astrocytes over here and get this volcano plot, which identifies some of the genes that are unique or upregulated in this tumor cell population that looks like astrocytes. So looking at these upregulated genes, we identified one particular gene that was very interesting, GRIA1. And GRIA1 is a gene that encodes for a GLU-A1 homodimer receptor. And this protein complex is actually important in the formation of synaptic junctions between neurons when neurons want to communicate with each other. And typically, the other members of the family are widely expressed in brain cells, including neurons, astrocytes, and oligodendrocytes. But GRIA1, being the only one that can make the homodimer protein, normally is only expressed in neurons and not in astrocytes or oligodendrocytes, as you can see from this violin plot. However, in our clone 1 tumor, it's pretty highly expressed at a comparable level to the neurons, even though they behave like astrocytes in the transcriptome signature, and they're actually tumor cells. So this was a very intriguing finding. And while our paper was being published, we also found 2 other pieces of literature that sort of supported our finding, in that other groups had done functional assays and showed that there were glutamatergic synaptic inputs to glioma cells to drive brain tumor progression, and specifically that identified the AMPA receptor, which is actually the receptor encoded by GRIA1, to be playing a major role in this process. And similarly, in this paper, they find a similar phenomenon regarding this AMPA receptor. So we think that AMPA could be a very important target for gliomas that hasn't really been fully characterized. There are these two papers that sort of look at it, but it's not really the focus. It's sort of the outcome of their findings. And I think it's interesting that we also find similar relationships of AMPA to glioma and tumor invasion. So given this, we think that it would be very important to be able to scale up our single-cell one-seq method to allow us to generalize these results to the general, to be more generalized to more patients. So we have also tried to scale up the mode of our investigation using microfluidics. So I'd like to briefly talk about this. These are our collaborators who worked with us on this project. As many of you know, there are many single-cell platforms to choose from when you're doing your single-cell RNA-seq. Many of you will be familiar with the 10x droplet-based method, and also, of course, the plate-based method that I and the previous speaker have mentioned. The sort of main comparison between the 10x and the plate-based method, or even the earlier Fluidigm C1 method, is that the plate-based method and the Fluidigm C1 method will allow you to do multiple additions into one compartment, meaning that once you have a single cell, you can do one reaction, and then once that reaction is finished, you can add more reagents to do the next reaction, which is what we do in the single-cell one-seq. However, with the 10x chromium system, it's not possible to do that. You put in your cells and a droplet is formed around the cell, and then you can't add any more reagents to that cell or to that droplet, meaning that it's a one-step reaction. And so this really prevents us from doing sort of more complex single-cell assays on this platform, including these kind of multi-omic assays, with the exception of ATAC plus RNA-seq. So to enable this multi-step but highly multiplexed single-cell assays, we developed this microfluidic chip called OneChip, and it essentially allows us to make multiple mergings of droplets in a massively parallel format. So this chip has about 18,000 sites on a single chip, and it will allow us to pair and merge droplets so that you can add reagents into droplets even after a cell has been lysed and reacted one time. So the maximum number of steps that we can do on this chip is 4 steps. And we have been working very hard to move single-cell one-seq onto this high throughput droplet platform. And very preliminary results on the DNA side, at least, show us that we can get copy number alterations from single cells from this chip in high throughput. You can see here, this is the whole genome sequencing profile that we get from the clonal results of a one-chip reaction, and comparing this to a mini bulk CNV result that we got from sequencing about 10,000 cells, you can see that the major copy number changes are detectable in both with similar resolution. So we're excited about this. It's not quite ready for prime time, but I will be very happy to share the results with the community once it's ready. Okay, I don't know am I running out of time? How much time do I have left?
5 minutes about.
Okay, 5 minutes. Then maybe I'll just do a very quick plug for the recent spatial omics work that we have done. 1 minute. Okay, this is just so, because I know that in the HCA community, a lot of people are doing spatial omics analysis. So I want to share these tools with you guys in case it might be helpful in your analysis. And I would really welcome feedback from anyone who uses it. So the first tool I want to very quickly introduce is SpatialScope. This is a tool that will allow you to get transcriptome-wide expression level information at single-cell resolution from your spatial omics data. So whether you're using Merfish or Visium or any of those very popular spatial omics platforms out there, you will know that often you're getting just a subset of the genes from the transcriptome from those platforms. So basically, you're getting some kind of a subsample of the cell's whole transcriptome information. But if you also have the nuclei image data, then our tool, and also a paired single-cell RNA-seq atlas from the same tissue type, then what we can give you is a single-cell resolution cell type identification, as well as for each of these single cells in here, a whole transcriptome level data set. And the way that this works is that we're leveraging a deep generative model. So basically, we're learning what the single cells look like from the single cell atlas reference. And then using a deep generative model, we can generate actually novel cells. We're not actually just aligning your data to an existing atlas, but rather generating the cell based on the data that you're getting from Visium or from MERFISH. So as an example, here is the Visium data from human heart that was published from Sarah's lab. And this is the Visium data at spot level. After using our tool, you can see that, first of all, it will tell you each spot what that cell type is. And then for each of those spots, you can get a whole transcriptome information. So after that, you can actually do things like, ligand receptor analyses or trajectory analyses over your spatial data. And this works also for MERFISH data if you have a subset of probes that you're looking at. And this is all using imputation rather than sampling. The second tool I want to quickly talk about is Stitch 3D, which if you have multiple slices of spatial omics data, this tool will, like this, fly embryo data that we have here, this tool allows you to stitch that data back together and into a 3D model, and that will allow you to, first of all, visualize that whole data in 3D, but more importantly, for example, with this brain data, once you've reconstructed that 3D reconstruction of the slices, you can actually then take virtual sections. So this is showing a real section, the real section data that's been piled up together, and here's the hippocampal neurons. And then once you have this reconstruction, you can actually then re-slice that section in a different direction and compute trajectories or gradients in a different dimension, depending on what your biological question is. So both of these tools have been published and they're available to download on our GitHub. If you do use it and you have feedback for us, I would love to hear from you. Okay, finally, the acknowledgements. This is my group. And of course, I've received a lot of support from my collaborators and also my funding sources. And just a quick plug that I am hiring for specifically evolutionary genomics and ecology with single cell capabilities. So please contact me if you're interested in this type of work. Thank you very much, and I'd be happy to take questions if we have time.
Thank you.
Hi, Angela. Great work. Congratulations, Jay here.
Hi. Thanks.
Hey.
Oh, hey, Jay.
Hi. Your one-seq shows a very nice co-expression or RNA and WGS or copy number variation. Have you benchmarked to existing infer CMV tools where you can infer CMVs based on gene expression, and what are the accuracy improvements based on one-seq? And also related is that I presume you're using TM5 or transposase initially to digest or segment your DNA. Are you seeing like a tech-seq like phenotype? Or are you able to look at heterochromatin regions where closed regions may not be so accessible by TM5? So any thoughts on that?
Yeah, thanks for the questions. The first question about infer CMV, we did do that checking. So the question that we had was, can we simply just recover the same measured CMV profiles if we just use infer CMV on our RNA-seq data? The answer is no. We don't recover that special clone that we found that looked a lot like astrocytes. It's completely missed by infer CMV. And some of the copy number changes that we measure are also missed by infer CMV. The reason for that, we suspect, is because no matter whether you're using infer CMV or the other tools that infer the CNV from the, or try to impute or infer the CNV from the transcriptome information, the problem is that you always need to input a reference as your baseline. So depending on what you input as the reference, the result or the outcome can change a lot. And I think depending on the sample that you are measuring the transcriptome from, the appropriate reference cell type for doing that inference can be different. And you may not always have that available on hand. depending on how you plan the experiment. So I think like a direct measurement will always be more accurate and more informative, I think. So as to your second question about the TN5, in our specific protocol, we use a proteinase K to do digestion of all the chromatin proteins and all that inside the cell or the nucleus. So we do not see any attack-like patterns. We very briefly tried, if we didn't do that proteinase K digestion, whether we would get that. It didn't work the one time we tried it, but it was not our primary focus, so we didn't push further to see if that would be possible. But given that 10x already has a very good attack plus RNA pipeline, we thought that our value would be somewhere else, so we didn't really pursue that route.
Awesome.
Yeah.
Thank you. Yep.
Thanks for the question.
Hey, Angela, beautiful work as usual. Ram from GIS.
Thanks.
So I was wondering actually about your spatial scope. So do you always need a matched single-cell RNA-seq data for your cell annotation, or could you just use a reference as well?
Yeah, that's a great question. We tested our tool using both like published references that were not generated at the same time, and also those that were generated at the same time. So definitely if your tissue is very specialized, it would be better if you have a paired or like a matched reference of the same type of tissue. But if not, it also works with a reference data set. So if you have, let's say, a typical organ tissue that you sequenced, and then there is the same organ that is already been cataloged by HCA or by CZI, those will work fine, and the tool works with it. You will get more accurate results if you have a very specifically matched single-cell data set.
Great, thank you.
Yeah.
Hi, Angela. Lovely work. Very excited about
you.
single-cell one-seq. One question is about the single-cell WGS. Can you comment on the price for that? And it seems like it's going to be pricey. If so, can you think of a selection criteria to select out particular cells, and if chips can be built to do that selection?
Yeah, it's always a concern, I think, when we do single-cell WGS, the price per cell. And our cost, like the cost of doing single-cell one-seq is not going to be higher than other existing single-cell WGS methods. In general, in our data that I just showed you, we sequenced each cell on average to 1 million or so reads. And we were able to get good quality data for both the RNA and the DNA side. We haven't tested how low you can go, but essentially the answer is, depending on what the size of the CNVs you're looking for are, you will need to sequence deeper if you want to see smaller or like higher resolution CNVs. We also have thought about developing, let's say you have a bulk sequencing data that's very deep and you know exactly which things you're looking for, whether we could do a selected amplification and go that way. I think it's viable based on our workflow, but I just haven't had enough bandwidth to throw into developing this particular application. And I think like one of the reasons I started a commercial entity is hopefully to be able to do that in the future, because I know a lot of people think that it would be useful. Yeah.
Thank you.
Thank you. Maybe one question from the online.
Hi, Angela. We have a number of people who called in online. So one of our online attendees has asked, how does single-cell one-seq compare to a recent preprint that uses 10x attack RNA kit with nucleosome depletion?
I haven't seen that preprint, unfortunately. I would have to look at it. So I'm sorry that I haven't seen that preprint, but yeah, I mean, now that it's been brought up, I will definitely go take a look at it. Yeah, I'm just not sure. I would have to look at it, yeah.
No worries.
Thanks for the question.
Cheers.
Thank you. So now I hand over to Liang.
So next speaker will be, thank you, Angela. Gonna be move to next final speaker. The final speaker from China, Wang Yu. It's about online. So please, it's connected.
Thank you for the invitation and also nice talk. Nice introduction. Maybe I have missed because of the internet. I'm Liang from BGI Research, and it's my great honor to be here and share some of our work using the spatial technology in the liver cancer. When we talk about the cancer, one word that came out is the heterogeneity. In my mind, the tumor heterogeneity is including the malignant cells themselves, and also the microenvironment involve a lot of the cell types, including the T cells, macrophage, and the other stromal cells. And also the tumor cells themselves and the other cell types, they will show in different functions during the tumor progression. And also in the last decade, the research have using the single cell tools to uncover the tumor heterogeneity well. We're using the single cell DNA sequencing, like the single cell whole exome sequencing, to understanding the genetic heterogeneity of the tumor and showing the monoclonal and the polyclonal. We also using the single cell RNA sequencing to identify the cell type and substates in the microenvironment, also to figure out what the difference from the prime tumor to the relapse and what the key factors and the key subtype to respond to the treatment. But when we use a single cell to talk about the cell connection, cell communication, we usually to based on the expression of ligand receptor pairs, and finally we will validate their coexist by the multiplexed IF data to support their exactly potential communications. In that case, it means the spatial information and to observe their coexist for the different cell types it's really important to talk about their communication. So in 2020, the spatial transcription have been highlighted by the net method, especially with the development of technologies. High resolution spatial technology at the single cell resolution, even the subcellular resolution have come out. And when combined with the spatial-omics data, I think for the cancer is really important. They could provide the direct information to figure out the crosstalk between the cells. Another thing is that we know that for cancer diagnosis, we use the slice, and for the spatial omics, we also use the frozen slice or FFP slice, which is exactly the object you see in the pathology diagnosis. So when using the spatial data, it's more easy compared to the single cell data. They are more easy to integrate with our pathology data, which may be more accelerate the procedure diagnosis. When we talk about the spatial technology, what they benefit or what they are showing their own advantage to do the cancer research, one issue will come out. Like in the cancer, in the tumor region, some of the structures are considered region really needed the spatial technology to investigate them, like the blood vessels and the tumor normal interface. And today I would like to share our work to using the spatial technology to understanding the tumor normal interface, which is exactly the area where the tumor cells investigate to the normal one, and they usually to think is the most aggressive area in the tumor. So to do so, we first collect the samples from the tumor core and the major area and the periphery tissue, as well as the lymph node, and did the single cell sequencing data using the DNB11C4 platform. This is a droplet-based microfluidic devices, which is the most popular strategy worldwide. As well, we get the spatial transcript data using the Spatially-resolved. And the Spatially-resolved technology is developed by my colleague and published last year. And this spatial this spatial technique is based on the DMB narrow ball sequence chip, and each narrow ball is 220 nanometers, and they have a lot of the have the DNA seq, and they in each narrow ball we have some barcode to label the location, as well have some oligos to capture the mRNAs when you have slides on there. And also the distance between the two nanoball is 500 nanometers. That's why we say this technology have the high subcellular resolution. That means when for a particularly cells of diameter of 10 micrometer, you can get 400 spots per cell. And well, if we also will have the image to staining the nuclei, so it will to do the cell segmentation based image. But for this study, we are sample the liver. So the major cell type is hepatocyte. They have much larger than the classical cells, is about 25 micrometers. So we used the bin 50, the 50 multiplied 50 bins, so to construct pursue single cell, because this data is produced 3 years ago. We didn't do the nuclear staining, so we to construct pursue single cells. And we also use the single cell data as a reference and use light to do the spatial cell annotation. You could see that the markers of a specific cell type identifying the spatial is how well expressed. So when we plot the distribution of the cell type in different regions, it's clear to say the macrophage and the fibroblasts are enriched in the tumor tissue, as well as expected the B cell is a major type in the lymph node. And when we focus on the major area where containing the tumor normal interface, we could see some microphages are enriched in the interface. Both in the HCC and ICC, the two types of the liver cancer. So to more comprehensive understanding the invasive front major area, we construct a segmentation approach to see their cell abundance and the signature and the dynamic changes in 2 directions. One is allow the tumor burden, and we can see the heterogeneity allow this direction, and another direction is away from the tumor burden in tumor site and the normal sites respectively. We can see the dynamic changes what happened. So it's clear to see that the immune cells are extremely enriched in the 250 micrometers, especially the macrophage. And also we validate this phenomena in another 100 cohort using the multiple IF. As also not just the macrophage showing the enrichment, but also we have checked immune checkpoint genes, which also showing the immune the classical immune inhibitory signal like the CD96, they are enriched in this area. As well, we have checked the signaling hallmark of the tumor cells. It will to see that immune escape signal also enriched in this area because of the considering the extremely enrichment of the macrophage and the signaling in the 250 micrometers wide. We define this 500 micrometers wide, including the 250 micrometers in tumor side and the normal side, both as invasive front. And there we would like to know which subtypes contribute to this enrichment of macrophage. And we do the clustering of all the cells in this invasive zoo, and we identify a subtype of hepatocyte. And they show in high expression level of SAA1 and SAA2, and they're plotted in the represented by the red dots. You could see that they are very close to the tumor burden, tumor board. And using the cell in the cell interaction analysis, we could see that the HEP1 could recruit the macrophage through the SAA and the SSA receptors, like the FPR1 and the TLR2. And when we to see also to confirm that the SAA1 positive hepatocyte and the SAA receptor positive macrophage are accurate in this area. And this phenomenon is not just in the primary liver cancer, but also validates in the metastatic liver cancer, like the colorectal cancer of liver metastasis. As well, when we validate by the multiplex IF, it could say that they have good correlation between the SAA hepatocyte and the number of the macrophage to support their recruitment. And also, when we think they have SAA1, SAA high hepatocytes, they show in the as the signature in the invasive zoo. So we would like to know what their association with the clinical. So to see that the more SAA high hepatocyte in the invasive zoo will showing worse prognosis. Considering the SAA positive hepatocyte will contribute to the immune suppressive microenvironment and also to association with worse prognosis. So we'd like to know when we target this subtype and to specifically knock out the SAA expression in the hepatocyte, then maybe will delay the tumor progression. So we construct a model with knockdown of SAA in hepatocytes. As it was clear to see that in the invasive zoo, total macrophage as well, the M2 macrophage showing the immunosuppressive phenotype are decreased in the invasive zoo. As well also the tumor size and the tumor progression is delayed in the SAA knockout model, not just in the primary tumor, but also in the metastatic tumor. That indicates the SAA hepatocyte maybe the normal cells may be a potential target in the liver treatment. So by using the spatial technologies, we have defined a 500 micrometer wide invasive zoo and also showing the crosstalk between the cells in the invasive. What is more important, we have highlight a normal hepatocytes. They could contribute to this immunosuppressive microenvironment and the tumor progression, and give our that one way to do the to select the treatment target cells, not just tumor cells, but also normal cells. Maybe the damaged normal cell maybe are potential target cells. And in the query, I have said that because we do not have the staining image, so we have used the merger beam to pursue a single cell. So we have added some figures to showing how the nuclear staining and showing the cell segmentation, and it's clear to see the crosstalk between the tumor cells, macrophage, and the damaged HCA positive hepatocyte in this invasive zoo. And so we would like to say single cell resolution spatial technology will really powerful to investigate the tumor environment in some concerning region, and will provide some new insights. And what I would like to mention, we usually talk about the multi-omics needed to be included for comprehensive understanding of the tumor. And also I would like to mention the spatial dimension, the 3D is also maybe considering. This is, we would like to mention the tumor body, how things separate usually to be thought is separated from the main tumor region. But when a paper published last year have construct a 3D information and found that the tumor body is not separate one; they are connected inside. So here is the example of the mouse embryo and showing the 3D. I really think this is fantastic. So I hope one day maybe for the tumor lesion we could also have some 3D pictures and to give the whole picture of the tumor lesion. And I would like to thank all my colleagues from BIJA Group, BIJA Research, and the Gene Bank, and also our colleague from Fudan University, Zhongshan Hospital of Fudan University. And also if someone will like to learn a lot more about the StillSeq, please just scan this and visit the website. I would like to also recommend this website, the Cloud-Stomics Take, because here you can get my colleague have done a lot of work, and you can get an example of StillSeq data, as also they have established some pipeline from the raw data to the matrix, and also the cell segmentation based on the StillSeq, and also some analysis tools, including the clustering and annotation and other things. So welcome to visit this website.
Okay.
And thank you very much.
Thank you, Liang. Thank you very much. Our schedule is a little bit behind, so I think maybe do not take a question. So thank you very much for your participation. I'm gonna close the session and then with thank to Dr. Arvind, and also we'll have a break for 15 minutes. Okay, thank you.
Thank you.